Hello students, hope you enjoyed the last lecture of molecular basis of inheritance. Today we are going to start next point, that is central dogma of molecular biology. This central dogma of molecular biology was proposed by Francis Crick immediately after James Watson and Francis Crick proposed the double helical structure of DNA. And according to this central dogma of molecular biology, the genetic information which is present in DNA flows from DNA to RNA and then from RNA to protein. And then proteins execute the actual characters of the organisms in the form of, say for example, enzymes, etc. So how this genetic information exactly flows, that we will study with the help of this flow chart. DNA produces its own copies by the process of replication. So DNA replicates itself, and then DNA can produce different types of RNAs, and for the expression of a particular protein, the RNA that is produced which contains the information is mRNA. So DNA produces mRNA and all other different types of RNAs with the help of the process which is called as transcription, and mRNA produces proteins, or it transfers its information in the form of proteins by the process of translation. So DNA produces DNA by replication, DNA produces RNA by transcription, and from RNA the genetic information flows towards protein by the process of translation. This was the central dogma, the central theme of the molecular biology. This is actually the central theme of molecular biology, which was proposed by Francis Crick. But later on, it was also discovered that RNA can also produce DNA. The genetic information can also flow in backward direction, not in complete backward direction. Proteins can never produce RNAs due to the degeneracy of the genetic code, but from RNA DNA can be produced due to the base complementarity which occurs between the nitrogenous bases of DNA and RNA. So this reverse transcription, which was not initially proposed as a part of central dogma by Francis Crick, was later on proposed by, actually it was identified and then given by Temin, and this reverse transcription is now considered as the part of central dogma. Because reverse transcription was initially identified by Temin, it is called as Teminism. And there are lots of examples of reverse transcription. The DNA that is produced from mRNA by the process of reverse transcription is called as complementary DNA or copy DNA, because it is copied from mRNA, that is why copy DNA, and because it is complementary to the mRNA, that is why this DNA is also referred as complementary DNA, which is popularly and shortly written as cDNA. We will talk about this cDNA later on also in biotechnology chapter. An example of one of the example where reverse transcription occurs is the HIV virus genome, जो humans में AIDS cause करता है. So when this HIV virus infects humans, its genome, which is RNA, produces cDNA by the process of reverse transcription, and that cDNA enters into the chromosomes of human, and then it starts further infection process to the humans. Okay, and there are some other examples of RNA viruses which show reverse transcription, but one thing you should remember that हर RNA वायरस में रिवर्स ट्रांसक्रिप्शन नहीं होता है. फॉर एग्जांपल टोबैको मोज़ेक वायरस है जो सोलानेशियस प्लांट्स को इन्फेक्ट करता है, तो इन वायरसेस में आरएनए से डायरेक्टली आरएनए बनता है, आरएनए से डीएनए नहीं बनता है. तो वहां पे भी कॉम्प्लीमेंटरिटी से कोई न्यूक्लिक एसिड बनता है, आरएनए से आरएनए बनता है, बट आरएनए से डीएनए cDNA वहां पे प्रोड्यूस नहीं होता है. रिवर्स ट्रांसक्रिप्शन इज नॉट शोन बाय एवरी आरएनए वायरस. Few RNA viruses only show the reverse transcription mechanism, and we use this reverse transcription mechanism in vitro in test tubes for our benefit also. We will study about those points in biotechnology chapter. How we exactly use this concept and this process for producing products and services beneficial to the human beings. Okay, we will study the next point now. Okay, now we will study the next point, that is packaging of double stranded or double helical DNA. Now the first question that arises is that why to package DNA? हम उस DNA को ऐसा linear ही क्यों नहीं छोड़ सकते सेल के अंदर या न्यूक्लियस के अंदर यूकैरियोटिक cells? To answer this, we will first have to study the length of the total DNA. The length of human DNA comes to be approximately 2.2 meters. If we take it into the meters. Yesterday हमने इसको देखा था base pairs में. तो meters में इसको कैसे convert करेंगे? Length of haploid DNA in humans, as we studied in the last lecture, is 3.3 into 10⁹ base pairs. इसको अगर double करेंगे मतलब total diploid DNA का length हम consider करेंगे तो it will be approximately 6.6 into 10⁹ base pairs. Now यहाँ पे 3.3 into 10⁹ base pairs है and diploid content में approximately 6.6 क्यों बोल रहा हूँ? Not exactly 6.6 क्यों नहीं बोल रहा हूँ? तो इसका reason है, in case of females we can say that the diploid content is exactly 6.6 into 10⁹ base pairs if the haploid is 3.3 into 10⁹, because in human females the haploid content is 22 autosomes plus 1 X chromosome and diploid content will be another same 22 autosomes plus another X chromosome. So 44 autosomes plus 2 identical X chromosomes. That is why the length of the haploid DNA content will be exactly doubled when we will double the chromosomes, when we will increase the copy of each of the chromosome in the diploid content. But in case of males, human males, this is, for example, the DNA, the number of chromosomes in the haploid content, 22 autosomes plus 1 X chromosome. But if we take the diploid content into consideration, then the another sex chromosome that comes into play is the Y chromosome in males instead of the X chromosome. Now the length of each of these 22 autosomes is equal to the homologous chromosomes of these 22 autosomes. So there is no variation in the length of the DNA here in case of autosomes, but X chromosome is larger and Y chromosome is very small. So if females contain 2 X chromosomes, their DNA content is little more than the male's DNA content because Y chromosome is little smaller than the X chromosome. That is why I am saying approximately 6.6 into 10⁹ base pairs. Okay, so this is the length in base pairs of diploid human DNA content. इसको अगर हमको मीटर में कंसिडर करना है तो we will simply have to multiply by the distance between the two successive base pairs. हमने कल DNA के डबल हेलिकल स्ट्रक्चर में देखा था कि दो बेस पेयर्स के बीच में 0.34 नैनोमीटर इतना डिस्टेंस होता है. इसको मीटर में कन्वर्ट करेंगे तो इट विल बी 0.34 into 10⁻⁹ मीटर्स. अब टोटल लेंथ का टोटल डीएनए का अगर हमको लेंथ निकालना है तो इट विल बी 6.6 into 10⁹ मल्टीप्लाइड बाय 0.34 into 10⁻⁹ multiplied by the actual distance between two base pairs, then it will come to be 2.2 meters. So total human DNA का length in any human cell, somatic cell, is 2.2 meters. And उस सेल के न्यूक्लियस का साइज, टिपिकल न्यूक्लियस का जो साइज है वो 10⁻⁶ मीटर से, मतलब इट इज 1 माइक्रोन मीटर. तो 1 माइक्रोन मीटर के न्यूक्लियस में 2.2 मीटर का डीएनए बिठाना है तो उसको लीनियर फॉर्म में तो नहीं बिठा सकते, उसको कॉइल करके, कंडेंस करके, पैकेज करके बिठाना पड़ेगा. ओके, जैसे टेलीफोन का कॉर्ड होता है तो उसको अगर लीनियर करेंगे तो उसका लेंथ बहुत बढ़ेगा, लेकिन उसको कम जगह पे बिठाना है तो उसको कॉइल करके, स्प्रिंग की तरह कॉइल करके उसको हमको पैकेज करना पड़ेगा. तो सेम फैशन में डीएनए को छोटे से न्यूक्लियस के अंदर अप्रोक्सीमेटली 1 माइक्रोन मीटर साइज के न्यूक्लियस के अंदर बिठाया जाता है. अब इसी तरीके से हम प्रोकैरियोटिक सेल्स में भी डीएनए पैकेजिंग क्यों करनी है वो देखेंगे। यहाँ पे भी सेम क्वेश्चन अराइज होता है कि द साइज ऑफ द प्रोकैरियोटिक सेल, वहां पे न्यूक्लियस तो नहीं है तो टोटल सेल में ही डीएनए बिठाना है, न्यूक्लियस में नहीं बिठाना है, लेकिन फिर भी द साइज ऑफ द प्रोकैरियोटिक सेल इन बैक्टीरियल सेल इज इन माइक्रोन मीटर्स, 1 to 5 माइक्रोन मीटर्स एक्सेट्रा, और एट मैक्स फ्यू मोर माइक्रोन मीटर्स, बट माइक्रोन मीटर्स से ज्यादा इसका साइज नहीं है। एंड इसका डीएनए अगर देखा जाए, फॉर एग्जांपल ई कोलाई बैक्टीरिया का डीएनए, तो उसका साइज बनता है 1.36 mm. So we have to fit a 1.36 mm DNA into a micron-meter sized cell, and that is why the packaging becomes necessary. In case of prokaryotic cell, the packaging is very simple. Because DNA is negatively charged, it can be associated with some positively charged proteins. So several positively charged proteins form a scaffold, a support-like structure, and the DNA is coiled and supercoiled on these positively charged scaffold proteins in large DNA loops. तो इस तरीके से ये लंबा सा डीएनए इस तरीके से कॉइल करके सुपर कॉइलिंग करके पैकेज किया जाता है in association with positively charged scaffold proteins in bacteria, in prokaryotes. But the packaging okay. Now we will study the next point, that is organization and packaging of DNA in eukaryotes. In case of eukaryotes, as I already said, that packaging of DNA is more complicated because the length of DNA is very much large in comparison to the size of the nucleus, and several different types of proteins are involved in the packaging process. Most important types of proteins that are involved in eukaryotic DNA packaging are histone proteins. These histone proteins are positively charged proteins. And why they are positively charged? Because they contain large number of basic and positively charged amino acids like lysine and arginines in their structure. So there are different types of positively charged histone proteins involved in the packaging of DNA. These are H2A, H2B, H3, H4 and H1 histones. 5 different types of histone proteins. This H stands for histone obviously, and H2A, H2B, H3 and H4, these 4 different types of histone proteins, they are present in 2 copies in each single unit of the packaging. और इस सिंगल यूनिट को न्यूक्लियोसोम कहते हैं। तो फॉर एग्जांपल ये अगर न्यूक्लियोसोम का कुछ पार्ट है जिसको हम हिस्टोन से बना हुआ ऑक्टामर बोलते हैं, तो दिस इज कॉल्ड एज ऑक्टामर बिकॉज इट कंटेन्स 8 हिस्टोन प्रोटीन्स। H2A के 2 कॉपीज इसमें रहेंगे, एक आगे यहाँ पे दिखाई दे रहा है, एक पीछे के साइड से है। H2B के ही इसी तरीके से 2 कॉपीज है, H3 के भी 2 कॉपीज, H4 के भी 2 कॉपीज। तो कुल मिला के 4 प्रोटीन्स के 2-2 कॉपीज, 8 हिस्टोन प्रोटीन्स बनते हैं और इसका एक कोर बॉल लाइक स्ट्रक्चर बनता है, इसको हिस्टोन ऑक्टामर बोलते हैं। And this H1 histone is also present in association with these nucleosomes, but they are not the part of histone octamer. Okay. Now, how exactly the nucleosome is formed and the structure is further coiled and packaged, that we will see here. So, each nucleosome is made up of histone octamer and a DNA molecule wrapped around it. Okay. Because DNA is negatively charged and histones are positively charged, DNA can get wrapped around the balls or the core-like structures of the histone octamer, and this complete structure involving histone octamer and the wrapped DNA is called as one nucleosome. So these are several nucleosomes in the packaging process. तो एक हिस्टोन ऑक्टामर या कोर के सराउंडिंग में DNA approximately 1 1/3 टर्न कंप्लीट कर लेता है, मतलब 1.75 टर्न कंप्लीट करता है। 2 कंप्लीट टर्न्स DNA हिस्टोन ऑक्टामर के सराउंडिंग में नहीं लेता है, इट टेक्स ओनली 1 1/3 टर्न। एंड इस 1 1/3 टर्न में जितना DNA रैप्ड है अराउंड द हिस्टोन ऑक्टामर, उसको हम कोर DNA बोलते हैं। उस लेंथ इन ईच न्यूक्लियोसोम is 146 base pairs, and एक हिस्टोन कोर से या न्यूक्लियोसोम से दूसरे न्यूक्लियोसोम में जब डीएनए एंटर करता है तो बीच का डीएनए इज नॉट एसोसिएटेड, जो बीच का डीएनए इज नॉट एसोसिएटेड विद द कोर histone, and that is why this DNA which links the two nucleosomes with each other is called as linker DNA, whose length is 54 base pairs. So when we consider the length of the DNA in a single nucleosome, we consider both these DNA molecules, the core DNA and the linker DNA. That is why we say that in a single nucleosome there are 200 base pairs of DNA. Okay, तो इस तरीके से बहुत सारे न्यूक्लियस के ऊपर डीएनए रैप होता है, तो मतलब डीएनए का कॉइलिंग हो गया, तो उसका जो ओवरऑल डिस्टेंस है 2.2 मीटर्स का इन केस ऑफ ह्यूमंस, वो अब थोड़ा सा रिड्यूस हो गया। एंड इस तरीके के स्ट्रक्चर को हम बोलते हैं बीड्स ऑन स्ट्रिंग स्ट्रक्चर। यहाँ पे स्ट्रिंग जो है वो डीएनए है एंड बीड्स जो है दे आर हिस्टोन ऑक्टामर्स और द न्यूक्लियोसोम्स। ओके, एंड इन दिस बीड्स ऑन स्ट्रिंग स्ट्रक्चर the diameter of the DNA, obviously जो DNA का actual diameter है वही diameter रहेगा, that is 2 nanometer, but इसका further condensation होके we will see in the next slide that the DNA further condenses and its thickness increases. Okay students, so we were studying the packaging of DNA in eukaryotic cell. And we studied the first part of the packaging, that is formation of nucleosome and formation of beads on string structure. So this is a general beads on string structure. This symbol indicates that the length of DNA in this direction is undetermined, matlab it can be several base pairs in length, and the DNA is extended in this direction also in several base pairs. We are just taking small portion of the DNA to understand the process of packaging. So once again, this is a beads on string structure where string is the DNA and the beads are the nucleosomes, and the diameter of the DNA is 2 nanometer or 20 angstrom unit. This is the first packaging level. In the second packaging level, these several segments of the beads on string structure, they come close together like this. They come actually very much close together, but for understanding the process I have shown some gap between these two fibers. Actually they come in contact with each other. Several such fibers come in contact with each other and they form the higher structural level of the packaging, which is nucleosome fiber. And due to several fibers coming together, the thickness of the overall fiber increases and it becomes 10 nanometer. So in the second level of packaging, nucleosome fibers are formed. Their diameter or the thickness actually is 10 nanometer. In the third packaging level, several such solenoid fibers, they come, nucleosome fibers actually, they come close together and they form the third level, that is solenoid fiber. Solenoid fibers का जो thickness होता है that is 30 nanometer. And again in the fourth structural organization, fourth packaging level, several such solenoid fibers come together. They increase the thickness of the DNA in turn, but the length of the DNA decreases due to such type of packaging. And the fourth packaging level forms the chromatin fiber. The chromatin fiber has 200 nanometer thickness. These initial structural levels, they are generally present in the interphase stage of the cell cycle. And finally, the fifth packaging level comprises of the formation of chromatid, where the chromatin fibers condense further, especially in the M phase. At the start of the prophase, the chromatin fibers start condensing and they get highly condensed at the end of the prophase, and we can observe chromatids in the prophase and in the metaphase of the M phase also. So highly condensed form of the chromatin fiber or chromatin is chromatid, and it is 700 nanometer in its thickness. And as we enter from prophase to the M phase, the two chromatids attached to the single centromere of a single chromosome are observable. And in the metaphase, in this way, the DNA is maximally condensed and it is present in its highest packaging structure or level. So this is how DNA is packaged in the eukaryotic cell. In the next lecture we will discuss about the next point. We'll stop here. Thank you.